to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 14th episode of the Weekly Bioanalysis. My name is John Perkins. I'm Senior Scientific Advisor for all things related to LC-MS technologies. I've been with KCAS for 9 months, but I've been in Bioanalytical for over 25 years. I'm here with my co-host, Dominic Guarino.
Hello, my name is Dominic Guarino, and I'm the Senior Scientific Advisor for all things related to ligand binding assays, biomarkers, and flow cytometry. I've been with KCS for almost 7.5 years now, but I've been in the bioanalytical space, similar to John, close to 25 years.
Today's podcast, as always, is brought to you by KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industries for over 40 years now. Dom and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding the podcast or any of KCAS's services. We're thrilled to have you listening again. As mentioned, this will be our 14th week recording the podcast, so if you're interested, the other episodes can be found at kcsbio.com. As a reminder, you can follow us on Twitter at the Weekly Bio and at KCS Bio. As we mentioned before, we kind of started this whole thing because of social distancing getting in the way of normal communication. Even today, Dom and I are in completely different places. I'm in upstate New York still. Dom is in Kansas City, and our producer is in a different part of the Midwest. This weekly podcast gives us the chance to connect with all of you, and we hope it's a chance for you to get to know us too, and even more, it's a chance for you to get to know KCS and the services we provide. As always, a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover, so please let us know. Then we'll wrap up and give you a little teaser for next week, letting you know what is coming in the future. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off, Dom is going to go over today's podcast topic before we jump into the news and resources section. Dom.
Yeah, John, excited to be here for week 14. Wow, podcast 14. Just wanted to share some exciting information with everybody out there. We've had over 13,104 impressions of our podcast and videos and over 2,200 hours of watched video. John, that's pretty cool. Glad to hear somebody's looking at it and glad to see that we're not wasting our time on this podcast, right? So today's episode is...
Can we guarantee they were sat in front of the computer watching, or maybe they put it on and walked away and it made it look like they were being busy?
Well, John, I didn't tell you I had my computer on for 2,000 hours. That's why it's at that number. No. Today's episode, it's part 4 of 4 on where does bioanalytical fit in the drug development process. There's the growing demand of bioanalytical method development and validation to understand the biology and mechanism of action of a drug. Most notably, those terms are pharmacodynamic and biomarkers. Podcast today, as always, we'll talk about news and resources. The main portion will be drilling down into that section. We'll talk about the frenzy that is the frenzy that is PD and biomarkers. We'll talk about the impact on discovery. We're going to talk about understanding the regulatory landscape. Most notably, the big term out there these days is the COU, or the context of use. And we'll help break that down. And hopefully you'll get a good understanding of how biomarkers are validated or done some sort of fit-for-purpose validation or even developed within the context of use in the bioanalytical space. John, what an exciting show we got going on today.
Yes, so let's move to the news. Obviously, as usual, we'll start off with COVID stuff. And over the last couple of weeks, there's been quite a few readouts on a number of vaccines, starting with the AstraZeneca and Oxford vaccine. As talked about, they've had success in clinical phase 1. So that's moving forward.
Yeah, it's definitely it's kind of emerging itself as the front runner. I think right behind it is the Pfizer BioNTech. It's nice to see people collaborating like that, but clearly that those two are kind of making their way. Although I think we're going to touch on one other that out of China that's kind of showing some real promise. And then there was also Novavax and Fujifilm recently in the news. So it's like a horse race. There's people emerging, but the AstraZeneca Oxford trial, I mean, they've got a nice track record. Really like what they're doing there. From what I've been reading, they seem to legitimately might be on their way. And then, they're talking about trying to get a vaccine to the general public in the November timeframe, John. And that, I'm as I've said on other podcasts, that seems pretty quick, but I would say with 100% certainty, there's never been this much historic effort and collaboration for a vaccine. So why not get something to market sooner, right?
Oh, sure. But then, it's also, I think for the AstraZeneca readout, basically they're saying it seems to have lesser side effects than the Moderna one, which is another of the front runners. And then actually today, Johnson & Johnson are talking about their vaccine candidate, which has come through an animal study, a monkey study, where it's showing good response from first injections. So they're, from the early readouts, they're saying that actually where the AstraZeneca and Moderna may need 2 injections to get things going, the J&J one may actually be good, really start things moving on 1 injection. Then that's given the good proof of concept for going into phase 1 for their, or backing up their phase 1 study.
Yeah, that's all the reason why these things take time, right? I mean, that, like you're touching on, trying to characterize T cell augmentation of T cell responses isn't something that happens overnight. Central memory, reoccurring, whether antibodies remain neutralizing. I think there's also, refinement of it to avoid having to do multiple vaccines, multiple injections. I think all of it is, everything you're saying is the reason why the standard vaccine, I think I read, was like something like 8 to 9 years is how long a
Yeah.
normal vaccine takes.
I think last time we talked, Merck said the quickest they got one to market was 4 years. So we're a lot, long way short of that. And then the other one, you mentioned Pfizer earlier, where they'd been talking in the early days about their principal candidate being one candidate vaccine. They've actually flipped to another. Of the 2 that they were working on, they've flipped from the one that everyone thought they would be proceeding with, so that the BioNTech thing is moving forward, but they've changed direction on that.
Yeah, not surprising. I think there's, when John, you've seen results from data that say, oh, well, this appears to be better, and better might be, a signal of, 100 versus 103. And you're like, well, I guess we'll take the 103, but that
Sure.
doesn't mean it's better at that, as time wears on. And I think, I hope that scenario I gave you is one that makes sense in your world, at least in the LBA world. We'll do signal to noise ratios, and we'll say, oh, hey, we'll select this. And then, as we go down further performance characteristics, we might have to pivot back to something because, this one isn't performing, a particular antibody. And so it's not surprising in the vaccine space that this is happening either. I mean, I'm still, as long as they're safe, I think that's the biggest thing. The way they're being made and the safety of them is ultimately what's going to determine. So, to date, the people think that, and there's been people that have been vaccinated since March with things, or maybe not quite that long. To date, there's been no adverse effects, and that's really the key. Now, if it's not effective, that's okay, so long as it's not causing any harm, which I don't think any of these vaccines are. Although I have some caution around mRNA vaccines,
Sure.
which is the Moderna one is.
Yeah, I mean, I'm not surprised that they make change midstream. I mean, I've supported a lot of projects in the past where a customer will come to us with 2 or 3 drugs, saying we want to test these all against a single indication, and depending on how the animal studies go, then we pick a lead candidate, and that goes from there. So I mean, even in GLP, I've seen that, right? They're not just in the discovery phase. So I'm not surprised by that pivot at all.
No, and then one interesting other company is that CanSino Biologics. They seem to really be making some headway, and that's a Chinese-based company, and their phase 2 data showed some antibody responses and T cell responses. So that I don't. It doesn't matter where it's coming from. I just hope we get a vaccine, and one that's effective. And then, of course, what's effective? 50%. They're
Yeah,
lowering the bar a
yeah,
little bit, I
yeah.
think, on what an effective vaccine is.
Sure, absolutely. Yeah, and obviously, just staying on that moving away from vaccines, but we know that a lot of companies are looking at different approaches and repurposing pipelines. And I think it was yesterday, Roche mentioned that their IL-6 drug Actemra had been tested against uh, severe cases of COVID-19 involving pneumonia. So they're in a phase 3 study, and it didn't meet its primary endpoint. So I assume that will now drop out of any sort of further development.
Yeah, I think there's also been similarly. I think Regeneron had a cocktail, and it's proven to be wishy washy on its efficacy of late stage COVID patients where they're really in that cytokine storm. What's interesting is there's a whole host of molecules being dusted off. Without we're working on several of them in our LBA side of things, and I want to be careful not to reveal too much about them. But some of them, for example, are I think I've mentioned one is an innate. Kind of if you can damper the innate immune response, you can definitely turn off the adaptive immune response, and that's one. The other one is a real interesting one. It's hormonal. And so, back before we jokingly said, "Oh, we should send our wives out because they're more resistant to COVID." Well, that they're now showing there is some. Um, so steroids are being used, but it makes sense that maybe a woman's steroidal hormonal like thing could potentially be used as a therapy as well, John. So I don't want to say too much about it because I'm not able to, but it's not surprising that they're leveraging that, finding that, hey, women might be a little more resistant. What is it about some of the
Sure.
hormones women produce that could potentially be beneficial, right?
Yep. Another direction to watch. Yeah, absolutely. Yes. Shall we talk about there was the study done about the rapid decay of antibodies in people with mild COVID-19. So it was actually measuring to see how quickly that declined in people's bodies. But again, like you say, it's not just about the antibodies, but that primary signal as to whether people have resistance to the disease that does not seem to hang around, and it sort of backed up what had been sort of talked about in previous podcasts.
Absolutely. So, I mean, this one, so sometimes you hear about just readings and reporting. This was actually like, coming from the New England Journal of Medicine. I think that sets it a little bit different when you think about the validity of things, although we thought the Lancet was pretty valid. But they clearly there is some, um, the end power here is decent. But again, there the it makes you to me it makes me think you might need to get more than one vaccine
Sure.
in
Yep.
order to have productivity, so I mean, I hope as the community starts to piece these things together, I hope we're listening as a community. I hope we're taking all of our data bits together. I mean, the thought of maybe just having a one shot vaccine might be unrealistic, based off of some of what you're seeing here. If you've naturally been infected and the antibodies are not long lasting, then well, just kind of makes sense you might need a couple shots to get a vaccine.
Yeah, I mean, it just really illustrates how much we're still learning about this. I mean, really, it's early days in terms of it emerged late last year, so we're still figuring out what this disease does and what it means for the body, etc.
Yeah, absolutely. I mean, I'm still fascinated by some of the. What I feel like there's more concern around, and we touched on this a little bit offline, is around heart issues and other lung issue, other organ issues that, even though you're asymptomatic, that could have some challenges. And I think that's really where I'd like to see more invested. Of course, we saw those publishing. You provided a couple weeks ago where they illustrated in 3-dimensional or 4D gels how the virus can attack certain cell lines and organs and stuff. So that, and then the whole clotting factor, all of that is where I hope we not in addition to the vaccine. I hope that's where we start to try to kind of focus things on in terms of therapeutics as well.
Sure. Yeah, I'm sure lots of people are paying attention to that, and don't need us to tell them. But yeah, I mean, obviously it isn't just the lungs. There's obviously a lot of different effects coming from this virus. So, yeah, the more we can look at these other organs, etc., the better.
But I wonder about that when 1% are on dexamethasone, 1% are on hydroxychloroquine, but 28% are still on, zithromycin, which doesn't do anything for a
Yeah.
virus. So, John, I hope you're right, but man, I mean, it seems like sometimes the whole field is always a little bit off what's out there. But again,
Yeah,
I hope I'm wrong.
I wonder how much that was the initial rush to get things going, whereas hopefully now there's a more systematic approach. Plus, the early phase, early trials were sort of based on that assumption that the lungs were the principal organ affected, and that if we fix the respiratory system, then hopefully we're good. But then the more we learn about it, the more we know that it is actually attacking other organs as well.
Yeah, you wonder about the liver and everything like that too. I mean, it's
Yeah.
just a matter. I'm sure it'll be years before some of that is truly understood. John, moving along, you had this really cool art, not cool, that might not be the right word, but a very interesting read around what Sweden did. So maybe you can jump in and share what that study is, or what that publication was
Well, that was
addressed.
actually a letter, and it is opinion contributors, but 25 Swedish doctors and scientists wrote to USA Today and say that Sweden went for herd immunity, and really, it does not make sense to follow that model because it hasn't worked. So they didn't really mandate sort of social controls. I read somewhere else that really Sweden's been as economically hard hit as every other country, so they didn't benefit from not imposing controls, but also their mortality rate is much higher than equivalent countries. So if you look at the Nordic region, Denmark, Norway, Finland, and Iceland are all much more. Their curves compared to Sweden are, Sweden's much higher. And then actually, as you read down through that, they sort of said we tried to get herd immunity, but we're still only seeing 10% of the population with antibodies, so they're way short of where you would need to be to have herd immunity. So I think Sweden's an interesting case to watch to see where they go from here. Do they go into a lockdown mode to mimic other countries, or how do they address it? But certainly in comparison to their neighbours, where I'd expect that they would do something similar, it's been very different, and they've come off much worse.
Yeah, that article just said flat out, we got it wrong, right? I think the prime minister of Sweden was like, we got this wrong.
Mhm.
I think certainly early on, you had to have some, I'm glad in particular, Kansas City, we were way ahead of the curve. We're still doing a lot of things that I think are more restrictive than most states, and happily doing it. I think we're fine with that. Although I see less masks being worn, although they're mandatory. People are kind of ignoring some of that, which is a little scary. But back to Sweden, I think the thought of herd immunity happening in 6 months was just naive. That's just not how it works. Herd immunity can take quite a long time. On a little bit of a side note, I heard some of those states that exploded. You heard they were kind of having. Remember, they had like measles parties back in the day. Like if one of your
Yeah,
friends
yeah.
got measles, you'd get together where they were having COVID parties and trying to infect each other. Man, I think when you on the surface that says, oh, that makes sense, right? But then when you hear, hey, you might have long-term heart conditions, that does not make sense at all.
No,
Like, what are you
that,
doing?
that blows my mind to do it with something where you had no cure, no vaccine, no real defense against it. To go and get yourself purposely infected, knowing that it has it has a high infection and a high fatality rate than many things. Yeah, I wouldn't do that.
Well, I
I'm
think that's
avoiding
huge,
it
right? I
for
mean,
that reason.
somewhere in there when you're I'll say under 30 or under 25, you're just like, oh yeah, hey, it's not going to happen to me. Or I think these were pretty 20 somethings, college age, even younger. But yeah, I think that's been stopped or ceased, or at least hopefully it was just more maybe like a real minor pocket of it, right? Um, so with that, let's talk a little bit about some industry assessments or things that are going on in the industry. I think that recently there's been some drop offs in some CROs, right? Most notably, Covance and Icon are reporting some significant drops in their clinical aspects of things. Although I read somewhere Covance Central Lab Services has made up some of that, and that makes sense because they're doing more COVID stuff. But any other thoughts on just the economic situation and kind of what's going on? I think there's You see pharma getting billions and billions, and then restaurants and hairdressers are
Yeah,
suffering.
I mean, it is amazing that was the one article I forwarded last week. I mean, yeah, everything is melting down except for the pharma industry. And as we've talked about in many of these podcasts, there's lots of money. We've got so many. We're seeing large numbers of companies going to IPO. There seems to be tons of investment, etc., etc. But then today I was reading, car makers are making massive losses. Both Volkswagen and Renault announced huge losses in the last quarter. And Volkswagen think they'll be okay annualized, and Renault are partly owned by the French government, so they're likely to still be here. But doesn't take away from the fact that they're losing they lost billions of dollars in the last quarter. So, I mean, if we continue like this, it's that's still going to be a long term impact to those industries. And yeah, I mean, restaurants, it's I still haven't been inside a restaurant to eat. I actually ate outside a restaurant for the first time, but we're still relying on takeout when we do eat out. Some of our friends have just opened a coffee shop, etc., and I hope they have the wherewithal to get through that first period and before I went ready for when things pick up. But yeah, I don't envy starting out in this current environment.
Yeah, I mean, I think as a family, we've gone out twice, and we've been open for months now, and I've had like three breakfast meetings, and then I have had, a couple um, drinks with friends. Very few, but like those places are still sparse, and one of them was great. You had to distance yourself. There was like, no seats, so you had to keep your distance. So, but I'm with you. I'm not even eating a whole lot of takeout. But back to the economic thing, there is always the great thing about a capitalist nation, and I'll promote that, is that it ebbs and flows and it goes elsewhere. So plastic makers, right? So now, everywhere you go in a drive-through, there's a plastic thing. You check out at Walmart or wherever you're at, there's a plastic thing. Those companies are skyrocketing. So and then also the mask makers are
Mhm.
benefiting from it. So I just think it kind of moves its way around. With that said, 50% of all restaurants are predicted not to come back. That's a real problem.
Yeah,
That's
yeah.
a big amount of our workforce that suddenly is going to have to get repurposed somewhere, or at least do something else. And no matter how much plastics we make and bend and so forth, that's a big gap.
Yeah, I
And
mean, and
it's
it's because
concern.
it's not just the restaurants. I mean, it's the supply chain to the restaurants. So to lose half of that is massive. Just it's like you say, it's not just the eating out in places, but it's all the growers, everyone else who feeds into that at those actual restaurants. I saw, was it yesterday? Pizza Hut are struggling as well.
No, well, the other, I think, I hope something else emerges to fill that void. I mean, like I said, there's some plastics, and again, those are flying off the charts. Your do-it-yourself places are still incredibly busy, and everybody's doing a lot of that. And then, even supply chain. You mentioned that grocery stores are, it's weird. Some of the grocery stores are sparser than I would like to think. So I think there's a lot, even though the restaurant industry is suffering quite a bit, and food chain supplies, they're going to have to repurpose some of where all that's going. Some, we still got to eat, people are eating, but it's maybe not going to a restaurant, it's a grocery store will pick that
And,
loss. So I hope
and,
it's like, but how
and,
fast it can recover is the issue. And then there's just this I'm cautiously optimistic about things, but then there will be some, think about sports, and people were making a lot of, now games are empty, we're playing baseball, no one's at the field. That's a lot of revenue loss for
Sure.
people that were relying
Absolutely.
on things.
Yep.
So
Yep. You just quickly talk about food, but there's like a particular loaf of bread that we usually had for years because we really like it. It's disappeared from the local grocery stores, and the earliest we're going to get it in is early August. So somehow their supply chain has got disrupted, and these are the weird ways that things hit you.
yeah, no yeast, man. Still can't find yeast. I have to buy it
Maybe,
online.
yeah, there you go. Maybe that's it. Yeah. Yeah. Yeah.
With that,
Let's move on.
exciting topic today, John. Right? We got part 4 of where does bioanalytical fit in the drug development process. And today we're going to talk about the growing demand of bioanalytical method development and validation to understand the biology and the mechanism of action of the drug. So that's like PD and biomarker work. And I'd say at least half of our work on the LBA side has fallen into this. And I'm pretty confident on the mass spec and most notably hybrid mass spec, you guys are growing like wildfire as well.
Yeah, I think for us, my earliest memory of like looking at biomarkers was probably around 2005, and we started off with a request to look at amino acids, and then yeah, we're seeing more and more small molecule. Obviously, it's much more on the LBA side, but there's still small molecule biomarkers that are becoming increasingly important. And yeah, we see regular requests for them. So you know that I was doing amino acids in 2005 at my previous company. But of course, we have a 24-plex assay for amino acids at KCAS, where different customers, but still really useful biomarkers for what they're trying to do.
Yeah, critical for any sort of probiotic nutraceutical, which are still those are still big time. Those are actually maybe benefiting from the COVID, right? That preventative health or improvement of health. Those amino acid panels are critical to the success of some of those companies. And for me, I mean, the word pharmacodynamics, I got introduced to it right out of 1993. I'm on an internship and walk by a room. I'm like, why is it dark in there? And they're like, oh, that's where the flow cytometer is, right? And so that was how we were doing PD markers for treating metastatic renal cell carcinoma patients with autologous T cell therapies. That was quite a mouthful, but that was my first foray into the biology, the mechanism of action, and then the big word these days is the context of use, right? And so that's really how we start talking about the regulated aspect of things. But before we move on to that, let's go in according to our agenda here, right? So
Yep.
we talked a little bit about the biology and mechanism of action. What are you seeing on the impact in the discovery space? Are people using PD biomarkers in the discovery space?
I think it's more the work is being done to support regulated, but because it doesn't need to be a fully GLP assay, then we support it in discovery. So it isn't really being done in the discovery realm. It's more, it's more a non-GLP approach to analysis, because all you need is maybe a qualified assay just to see is is there an effect of the drug that we're looking at, and maybe longer term it goes to a more validated approach. But certainly we do see a mix on that, and so our discovery group does get biomarker requests.
I see. So it's not necessarily in the drug discovery space. It's just more and more people are doing what we'll call discovery biomarkers by LCMS, and that makes a lot of sense. I mean,
Yeah.
almost all of our preclinical work is what we'll call non-regulated or non-GLP work. So maybe that's a good way to segue into like how we at KCS tackle the question around the regulatory landscape for bioanalytical analysis of biomarkers, right? So I mentioned the context of use. So to me, there are three main buckets. You've got regulated, non-regulated, and then developed and characterized. And so based on the context of use, for regulated stuff, we have two categories: a fully GLP validated method or a GLP fit for purpose validated method. And to me, it's like if you're going to use it as a primary endpoint, you're going to make some decisions around, is this person going to be dosed a different way? Are we going to include them within the trial, right? Are we going to dose them? That's going to require what I would consider a full GLP validated method.
And I
One,
would completely agree with that. Yeah,
yeah,
that's exactly the way I would approach it too. If that's a primary readout, then you want to have as as tight an assay as possible. So you really want it to be GLP validated, and yeah, it's you're way beyond fit for purpose at that point.
yeah, and then you get into like there's actually a GLP fit for purpose validation, and that can still be a primary endpoint, but we tend to do it for things like enzyme activity assays or big multiplexing panels that they're not often used as endpoints there, but like the sticking along the enzyme activity assays, or even in the gene therapy space, some of those pharmacodynamic markers are definitely, they need a QA review, they need to be fit for purpose validated, but they can't hit a GLP validated state because they just lack the right tools to do it. Meaning, what's the gold standard for some of those methodologies? You can't do the true GLP validation according to some of the guidances that are out there because you lack the proper tools to do it. So, that's some of the things we can help, consult on and certainly interpret with our clients. But those in the regulated space, there's that GLP validated, GLP fit for purpose validated space. And I think for me, it's primary endpoints and your secondary endpoints. If you're going to, again, if it's a secondary endpoint and you want to be like stratifying your data and doing some real critical analysis using that pharmacodynamic, or it's again impacting how you might use your dosing and PK and so forth, then you need a GLP fit for purpose.
Yeah, and some of it for us might be what matrix you're actually in. So if we're looking in plasma at those markers, that would probably be a GLP validation. But then if you get an additional study that's tissues, it's only a few samples, that might just be that a fit for purpose validation / qualification is good enough to support the tissue analysis looking at those same markers.
Oh, that's a great point. Oh, you are spot on there, John. When you go from things like plasma, serum, whole blood, all of them validatable for sure. Even like CSF, sometimes you can validate, but that's a matrix sometimes where you might use a fit for purpose. And then tissues, absolutely, because part of it is you still got to do a BCA. You got to homogenize them. You got to do a total protein measurement, and then you got to do your PD measurement. Well, that makes it pretty hard to do a true GLP validation of that according to the interpretation of those performance characteristics, because you got multiple
Sure.
data bits and even where are you spiking in your controls and so forth, right? Like that becomes somewhat of where's your reference standard if you're trying to validate that?
Exactly. How can you guarantee your standards are behaving the same as samples? That has been debated for years.
Yeah, and so then we got that regulated space, and then I think the bulk of the biomarker space actually falls into this non-regulated. And what I mean by that is a non-GLP fit for purpose validation. And that term is qualified. And the field goes back and forth, John. I sit on a lot of biomarker committees that we're trying to eliminate the word qualified so that we can be clearer that the marker's still validated. It's just a non-regulated fit for purpose validation. Because essentially
Sure.
you're doing most of what a validation would go through. It's just the number of accuracy and precision might be a little lower. Some of your criteria might be a little wider. And then, of course, you're not getting that QA regulatory component of it. But the reality is the method is, GLP-like, right? We don't like that.
It's still as good as you can make it for for the purpose of what you're doing. Yeah, absolutely. I mean, there's been an awful lot of debate in EBF, etc., in terms of fit for purpose validations for biomarkers on the LCMS side as well as the LBA side, and really trying to push that language rather than just qualification.
Yeah, and what we get a lot, and then there's a third bucket, and that's what we call developed or characterized. And this is the one that gets a little more, I don't know if controversial is the word, but a little more, I'm going to say wild, wild westish. So some people will say, hey, just take this ELISA kit and run my samples. We do not do that. We don't like doing it. We will push back. What we really like to do is at least run the kit once. And you could say, well, Don, what do you really learn with running the kit once? Well, quite a bit, right, John? You can learn a lot from the kit running it once. It gives the technician or scientist an opportunity to kick the tires on it, right? You can see if the lower limits or upper limits are going to behave the right way. You can make some QCs and see how they recover. And it gives you the ability to say, hey, client X, this kit, we ran it once. Hey, it looks great. Let's go. Or, hey, you know what? We better invest another couple days in this kit before we run your samples. So we do have even grades of development. And then that's probably the bare bones we'll do is 1. And then we go up to like 3 to 5 development days. And we call that kind of characterized because you can start to get some accuracy and precision around the kit.
Yeah, I mean, I think as bioanalysts, what you always want to give as good a data as you can get, as you can to the customer. I mean, this is similar to what we do within discovery, where you've got the 3 different research grade assays. And although there are definitely circumstances where the RGA1, which is just basically pull a method and run it, would apply, you want to know that the assay is going to do what it's meant to do. So I always much prefer to go with that next stage where you do at least a single qualification run. That even though you're using generic internal standards, you're using fairly generic conditions, but you still want to show that you're giving robust data for that experiment, whatever it is. I mean, I know we do the research grade 1 assays, but I have to say, as they sort of make me sort of twerk a bit, I like a little bit more
Outside your
backing.
comfort level, you're a PK GLP scientist, John.
Yes, exactly. Yes, I can deal with it, but it doesn't always. I always want to have that little bit more backing to it. But I agree with you in terms of you want to be sure that you're on reasonably secure ground before you start touching samples, because samples are precious. You don't want to waste them, and also you want to know that if you've used them, the data that you've given is as good as it can possibly be based on the needs of the assay or the experiment.
Yeah, I mean, in the LBA space, typically we're talking about kits. So the manufacturer should have done their due diligence on the kit, and that's not always the case, right? And when
Sure.
you do enough of them, I think last year we touched like upwards of like 100 different kits we looked at. So when you do enough of them, you get the ability to quickly assess them over. And I've had people challenge me on this. What are you going to learn in one day? Well, you do. It's like riding a bicycle, John. Like you can learn quite a bit about how that kit, like if it's not just a simple capture, put the sample, the plate's coated, you put the sample on, you detect. If it's got all sorts of if it's a competitive and some sort of competitive ELISA, and that I won't go into great detail. Maybe that's a whole nother podcast. But there are certain configurations of the ELISA that immediately we at times will say, hey, this is going to take 3 days. We can't do it in one. But most of them are pretty straightforward. And if we look at what the manufacturer has done, you talk to the manufacturer. The context of use of the client is exploratory, truly just exploratory, wants to get a sneak peek. Then doing 1 day assessments and understanding the minimum required dilution of the sample, that's critical. Understanding if we can dilute it, if the sample comes back AQL, we can do something that ensures it can be, the dilution linearity. We take a look at some of that. I mean, you got a lot of real estate in a 96 well plate to do quite a bit. And frequently we end up like, at least in our world, sometimes we'll budget a day, but we'll do it twice just to get a better handle on the kit. We typically budget to have a couple kits just to run it. So, that's kind of where a nice comprehensive look at, biomarkers. This is a growing space. As I mentioned, we touched at least 100 different ELISA kits last year. And, you can, as you do more of them, the team has really gotten a good ability to assess it pretty quickly.
Yeah, I think you definitely have more challenges on that front than we do in LCMS, but I understand wanting to make sure that you set the foundation for what you're doing for the subsequent experiments. So, is there much more to say, or shall we move on
No,
to
I think we
feedback
can. I
and
mean,
questions?
I didn't touch on multiplexing and flow cytometry. They all fall into the biomarker space, but the objective here was just to give a little bit of background around the landscape around the biology and mechanism of action, and then some of the regulatory things and challenges. And I think we did a nice job of touching on how bioanalytically, the buckets that it falls into: regulated, non-regulated, developed, characterized.
And
And
I,
it's
I
all
think
based off of, how
I,
much, what your context of use is.
I think flow cytometry is a whole nother podcast by itself.
Well, that one, it doesn't have as many buckets as you can imagine.
Sure.
It's
Yeah.
not everything I just said. And similarly, multiplexing gets more challenging because, if you got 10 analytes in there and one's misbehaving, what do you do, right? You can't, you opt, so and we've fallen down this rabbit hole. You optimize one and boom, another one falls out, right? So now you're just robbing Peter to pay Paul. So multiplexing becomes a
Yeah.
whole nother layer of like expanding the acceptance criteria from say 20% to maybe upwards of 30. And then it becomes I don't want to call it I don't know if a shell game is the right word, but your lower limit of detection or quantitation might vary from plate to plate because, if you got 10 analytes in there and one of them falls out at the lower limit, you can't rerun that whole plate. You just got to say, instead of 5 picograms, it's 50 picograms, and we're not going to report data below the 50 mark. Of course, everybody wants to see what that data bit is, even if it didn't pass. So there's all sorts of ways to handle that as well. And again, that's a whole nother podcast, John.
Yeah,
So the managing of
multiple
multiplex data,
analytes.
cytometry data is vast.
Yeah, multiple analytes are definitely one of our challenges. I mean, it's, for us, we want to try and get all analytes in one assay because it's more efficient and you can get to data quicker, but you can't ignore the chemistry, and sometimes it just isn't possible. And it's that getting that balance between all the different needs of the analytes is always a challenge.
Yeah, nice, John. Very nice discussion. Let's go on to some feedback, and looks like we got a couple. People missed us, John. That was, I on a sidebar here, I had a few people ask, like, I had a friend like email me and was like, "Hey, what's going on with your podcast? Did they cancel you?" He kind of jokingly said. I said, "No, we just took a week off," and so it looks like we've got a couple things though. John, you want to maybe take
I'll
the first
start.
one?
Yeah, absolutely. First of all, Lauren says, "Thank you for what you do. I have missed listening the last week or so. Looking forward to your next episode." Yeah, thank you, Lauren. We'll keep this going as long as we can. We've said it before, and this is episode 14, and I think we have about 9 or 10 more topics that we want to talk about at least, and obviously other things will come up in the meantime. So we've got plenty of stuff to talk about.
Yeah, thanks, Lauren. I actually missed it. I crave it. We got some feedback here from Devin. Devin says 13 episodes. You guys are legit now. I like the episode about CROs and vacation breaks. Keep up the good work, gentlemen. Thanks, Devin. I think as I'll tell you what, if not everybody's cut out for the CRO space, we are 24/7, 365.
Yes, it's that ability to deal with the stress and actually not just deal with it, but enjoy it. And I think there is definitely a fine line on that one.
Well, that'll do it for the feedback. Like I said, I did have at least two colleagues ask, and then a friend was like, "Hey, what's going on?" So it's good to know there's somebody listening, John, and we're just not talking to each other here, right?
Exactly. So yeah, that's wrapped up number 14 then. So next week we do plan to talk about an entirely different topic, since this has been a 4-part over the last 4 weeks. We'd actually like to talk about, we mentioned validation, but we actually want to talk about the purpose of validation, how it gives confidence in bioanalytical data. I think when you first start in a doing this work, you don't necessarily appreciate what those validation experiments are telling you, but it's a really useful part of the process. So we want to focus on that next week.
Yeah, fascinating topic, John. I assure you, the word validation is interpreted very differently from person to person, from company to company. And being in the CRO space, we tend to have to manage it because large pharma has an understanding of what a validation is. And then you've got people over here who are bootstrapped and they hear the word validation and they think another thing. So it'll be a great topic for sure.
Yeah, we've also certainly we've moved on a lot since the days where dealing with multiple customers, all the validations were different. With industry bodies talking about validations, at least we're much more uniform in approach. So yeah, that'll be good to
I
touch
still
on.
think there's a CLIA laboratory and a GLP laboratory have completely different understandings of how you validate a method. And I think that's troublesome, especially in the COVID testing space. Most of your antibody COVID tests are going to reference laboratories. And, that to me, the way they validate some of those things is part of the challenges we're seeing with what's really positive and asymptomatic and so forth. But
So
John, with that,
there's a subtopic for next week. Let's
yeah,
not spoil it
yeah, a whole
now.
other. So what you got going on this weekend? Anything fun?
This weekend, no, nothing really. It's sort of quiet. The only thing that we're looking to do, our backyard has just been a big square of grass for the last 20 years, and we're actually planning to do something different with it. So we've been sketching what we want to do, and then we've got a gardening company who are going to come and dig out beds and things like that. So that's probably the most exciting thing we're looking at. yeah, I think that's probably it. Oh, we've had the wedding I was hoping to go back to in the UK in October that I've formally pulled out of, so I'm not going to see my nephew get married, which is a bit of a shame. And then one of my son's friends is also, they've had to postpone their wedding, so our social calendar that looked interesting a few months ago has now become zero again.
Yeah, back to takeout and wine on the porch, I guess, right?
Yeah, absolutely. Yeah.
Yeah, I have the fun endeavor of I just purchased the materials for a new deck. This is quite an expensive endeavor. I've never invested in a deck. I can't believe how much I just paid for deck materials. And come Monday, gentlemen will be here to knock down the old one and start putting that up. So that'll be some fun. I don't have any big plans this weekend. The most challenging thing I have is my license is expiring. So to shave or not to shave is the question I have now. That's, I got a tough life, John. These are the things that are going on. But before we sign off, I was going to go to Boston next week. But I don't know if you saw, Boston mandated come August 1, anybody traveling into the state had to have a test and had to be quarantined. So that unfortunately, my social calendar, like yours, has been put on pause. I was going to go visit my ma for her 75th birthday. My niece was turning 15. I'm turning I got a birthday coming up. We all land right around the same time in August. So we were going to all get together, and that's kind of been put on pause, unfortunately. So
Yeah. So
I'll be around next week. I'll be here to host with you, John.
We've just gone through the July birthday flurry in our family. It's my sister-in-law today, but yeah, there's been a bunch of us. Yeah, and talking of licenses, my wife's driving license expired, so she was without for quite a few months. And then she finally got it, and I think it was the day that she got her new license, she was rear-ended. So
good thing, right? Hey, just because I can't go a podcast without talking about any sort of food, John, and didn't mention CAR T cells either, but or gene therapy. Maybe I did, but the pizza's been put on hold. Too hot. That's my. So lately we've been that's been on hold for me, but I'm sure as we wear into later part of August, I'm going to start making some pasta sauce again, and that'll make me want to make some pizza. But got to talk about pizza time. That's a big shout out.
It's tough, the cooking when it's like hot and humid, and yeah, we're sort of trying to find different ways around it so we're not getting overheated in the kitchen and stuff.
Yeah,
So,
well, I told my wife I'm going to put the gas grill out front, and she really shot me one of those looks of like that made me go, "I'm just kidding, just kidding." So, all right, I think that's a wrap.
So number 14 over and done. So we look forward to number 15 next week.
Wow, bona fide. All right.